Hello and welcome to my presentation for the Undergraduate Research Conference 2021. My name is Liam Sheedy, and I'll be going over my project, Transforming Camelina Sativa with PA-DGAT1 and PA-PDAT1 from Persea americana for increased oil yield and omega-3 synthesis. So what are DGAT and PDAT? These are two genes involved in triacylglycerol synthesis in both avocado and camelina. DGAT stands for diacylglycerol-acetyltransferase, while PDAT stands for phospholipid diacylglycerol-acetyltransferase. This project looks to take the avocado homologue of these genes and transform them into camelina. And moving forward, we'll note the avocado variants as PA-DGAT and PA-PDAT. Both of these genes are used in the diacylglycerol to triacylglycerol catalyzation step. DGAT1 catalyzes the formation of ester lineages between fatty acyl-CoAs and the free hydroxyl group of diacylglycerol, while PDAT1 transfers acyl groups from the SN2 position to the diacylglycerol position. These are two separate pathways that can be used to finish out tag synthesis, and both have been shown to be rate limiting in some regard. Research previously done in our lab has shown that the overexpression of DGAT1 in camelina, along with another gene GPT1, positively enhances seed oil yield and can be used to increase the overall oil quantity of seeds. A little bit of background about Camelina sativa. This has been used in the past as a dedicated biofuel crop because its seeds have an oil content between 30 and 40%. It's got a low growth cycle and can be used as an annual crop with low agricultural input. It also responds very well to drought conditions and can be easily transformed. If we can increase the amount of oil the seeds produce, it could be used as a biofuel crop, and if we can make the oil healthier, it could be potentially be used as a cooking oil. Persea americana is a plant more of you are probably familiar with. Avocados, they have a very limited growth range, require extremely large agricultural inputs as well as specialized equipment, and refining avocado oil is very inefficient. It does have a delicious mesocarp, however, which is what you guys like to eat, as well as a healthier oil. So what makes avocado oil a desirable product? It mainly has to do with its high proportion of polyunsaturated fatty acids and phytosterols. It's very similar to olive oil in composition, with high levels of omega-3 fatty acids and lower levels of omega-6 fatty acids than something like corn, canola, or camelina oil. This high level of omega-3s when compared to omega-6s has been associated with cardiovascular system benefits, including anti-inflammatory effects. So to compare and contrast camelina and avocado oil is a lot higher in palmitic and oleic acids, much lower in stearic acids and linoleic acids, as well as linoleic acids. And these are the areas we're looking to alter camelina oil composition. So the goals and hypothesis for this project, we want to transform Camelina sativa with PA-DGAT1 and PA-PDAT1, and then we want to analyze the oil and see how that changes both the composition of the oil and the total oil yield. We're expecting the plants transformed with DGAT1 to show increased oil yield, similar to the experiments done with the Camelina variant of DGAT1, and we're expecting the plants transformed with PA-PDAT1 to have oil richer in omega-3s and lower in omega-6s and more closely resemble that of avocado oil. This can help lead to a healthier cooking oil or a crop that can be used for biofuels in the future. Our materials and methods. The gene constructs were put into Pcambia from a collaborating group at the University of Eastern Tennessee. Thank you very much, University of Eastern Tennessee. And here are the constructs. Here's the DGAT construct. And here's the PDAT construct. The important things to note are the DS-RED vector. This is what we're going to use to screen our seeds, as well as the beta-conglycin promoter. This is a seed-specific promoter, which will make sure that our genes are only expressed in the seeds, which is where we want oil synthesis to be happening. So how do we get these genes into camelina? We take the gene construct and we transform that into agrobacterium. Agrobacterium is a really cool bacteria that causes tumor in plants through horizontal gene transfer. And we can utilize this biological mechanism to put a gene of interest into camelina at a rate of about 1%. So going from A to D in this figure, A shows what camelina looks like right before we transform it. Nice little bright yellow flowers that we then dip into a solution that has the transformed agrobacterium. That's what you see in B. That bubble is actually a vacuum. Under vacuumous conditions, you get about a 1% transformation rate. We then raise the plants all the way up to maturity, harvest them, clean the seeds, and then we look at them under a red fluorescent light to see if they glow red. After that, we can then check the plants for a variety of different characteristics. So screening and raising our homozygotes. You take the first plant that you transformed and you comb through the thousands of seeds, hoping to find 15 to 25 bright red seeds, quite literally like finding a needle in a haystack. You treat those seeds with a lot of care and you bring them over to the greenhouse and you plant them again. Raise those seeds up, and what we're looking for, sometimes you get homozygous seeds, a lot of times you'll get heterozygous seeds, and we're looking for 75% heterozygous seeds. That's what we have on the right here. On the left are wild type seeds, and you can see they're glowing nice and red, and that's what we're looking for. We then take seeds from the lines that show 75% heterozygous. We plant those again, we raise them up, harvest the plants, and then we get homozygous seeds that are showing 100% DS red expression and are nice and bright. Additionally, we need to check the expression at different intervals of seed development. These are the primers we'll be using for that through qPCR. We'll be checking at 7, 14, 21, and 28 days after flowering to see how that impacts the expression rates of these genes. So now to get on to some results very quickly. Unfortunately, COVID has gotten in the way of a lot of these results, and they're not ready in time for this presentation, but some are. So we'll start with the DGAT results. We do a lot of seed weight data. If the seeds weigh more, chances are it has more oil in it. So this is the 100 seed weight from the T2 Camelina seeds that were showing 75% expression. And we can see only one of the lines was showing a lot of potential. So we went ahead and we planted all of those seeds to try and figure out what was going on. And then we have our T3 seeds here. These are averages of all of the lines that got planted. And we see that line 7 and 10 were actually statistically had more biomass. And this is really promising. It shows that potentially they have a higher concentration of oil. And that was backed up by our nuclear magnetic resonance data. We ran this on lines 3, 5, and 7, and we found that all actually increased the oil concentration between 1 and 2%, but lines 5 and 7 were statistically significant by a good bit, and that's really exciting. We're going to go over the PDAT results now. These are extremely limited because of COVID delays. We just have seed weight data that shows a promising trend with most of the lines measuring higher seed biomass than the control. So what do these results mean? The data is promising to begin, but wildly incomplete at this moment. The transformed lines do show an increase in seed biomass, which indicates greater oil yield. And the NMR data backs this up. And this is really promising and really exciting. We still need to do a variety of assays. And this includes the expression analysis to check how the expression of these genes alters over seed development, as well as liquid gas chromatography to see what the actual composition of this oil is. And then NMR data from the PDAT seeds also needs to be taken, and that will all be done at some point. So with this initial NMR results and seed data, it looks like, at least in the DGAT seeds, we do have an increase in oil yield that has been accomplished through this transformation, and that's really exciting. Due to the differences between the two proteins, we actually need to look at the contents of the oil, though, and analyze that to determine if the omega-3 and omega-6 concentrations have changed. It looks like we might be able to use this to better Camelina as a biofuel crop, but we do not know about its oil potential right now for cooking or health benefits. So that's the presentation. Thank you so much for listening. I would like to thank Dr. Omprakash Dankar for allowing me to work in his lab. It's been a wonderful experience. I've really enjoyed it. I'd like to thank all the grad students, Ahmed, Kripal, Sudhir, and Jess, as well as Helen for helping me water my plants. I'd like to thank Dave and Chris in the greenhouse, as well as my parents, grandparents, and my wonderful friends. Thank you so much.